Hi, everybody. Welcome back. I am incredibly excited about this very interesting conversation we are about to have with Dr. Tomas Duraj currently from Dr. Thomas Seyfried's lab in Boston, but also has an incredible background. He's a medical doctor, so he's very accomplished at the bedside, but he's also a researcher and has a PhD on that level, so also equally as accomplished at the bench. And in my years of knowing Dr. Tomas, I have gotten to understand that he loves both equally and that he's always trying to find a way to kind of bring the two together. So I'm excited about where this conversation is going to take us today, some sort of heady discussions around metabolics in the cancer space and what this means and where we're going to go with it. So welcome, Tomas. I'm really glad that you're here.
Thank you, Nasha. Thank you for having me. It's a pleasure.
Absolutely. So first of all, you are, like I said, no stranger to both patient care as well as sort of peer-reviewed publications and the laboratory side of things. Tell us a little bit about what got you involved in this space of metabolic oncology. What drew your interest and your attention in this arena?
I after doing my medical degree, I was sort of interested in kind of going to it like a layman from outside to the research side about these new concepts in metabolic approaches to cancer treatment and to other diseases in general. And that was in the time where ketogenic diets were kind of having a resurgence in the mind share, and so I was trying to understand how these things work at a physiological level, and there is a lot of biology behind it.
Yeah.
And I was also struggling on a personal side, also struggling with being a little bit overweight, and these concepts naturally fed into into weight loss and all these things. So I kind of came into it from like a side opening, let's say. But then I the focus on the research is really what drew me in, and it's where like a lot of open questions remain for metabolic treatments, especially for cancer, and that's kind of where my focus is right now.
I love this because you talk about, kind of what we're going to chat about today, kind of this overarching piece here is this, there's a lot of differing opinions and interpretations of the data, number one. So we're going to kind of unpack that. Even the way our experiments are designed may be inherently flawed and maybe areas of where we can improve upon or just have an understanding of that, where we're prioritizing research today in the cancer environment and maybe what might be hopeful in the future of where we could put that. And then the other piece is like, how do we make this moving from kind of an intellectual exercise into a clinically relevant exercise? So I know that those are areas that are very near and dear to you because as a researcher, you get put into a camp, but as a clinician, you have a different understanding and a different need for this information. So can you talk to us a little bit about these discrepancies and where there's sort of holes in the narrative and in clinical application?
So I think one of the issues that we have in the field in general, and both on the research side and on the clinical side, is that right now there's different interpretations of what kind of metabolic cancer as a metabolic disease is, and the metabolic treatments, how they would be structured, and this comes from kind of an underlying theoretical background or theoretical framework that you would look at it. So like the traditional perspective in the research field is that we have to differentiate. There's cancer as a metabolic disease in the sense that cancer has like a hallmark of metabolic reprogramming as a genetic hallmark of cancer as a consequence of the mutational burden. And then you also have cancer as a mitochondrial disease, as you know, originally proposed by Otto Warburg at the beginning of the 20th century, and then in recent times continued by Dr. Thomas Seyfried and a couple of other research groups around the globe. And this understanding, it is kind of the foundational background and perspective. It's important because when you go to design treatments and when you even like experiments in the lab, and you tailor the clinical practice with different endpoints or end goals. And of course, there is also like a third camp, I would say, or third perspective where, and I would say this is the most prevalent one in the research world, and that is that cancer is everything. I don't know if you've seen the latest update to the hallmarks of cancer. It's I think published now in March in Cell, and it's titled "Embracing Cancer Complexity." So it's like now everything is cancer, every single kind of observation that you have. And I think this is missing the point because they mention a little bit about precision nutrition and fasting mimicking diets and PA3 kinase inhibition together with the ketogenic diets as a part of the dietary aspects that determine cancer carcinogenesis and cancer development. But then like everything is put under the umbrella of cancer.
Right.
And that is fine. That's like a reductionist approach where, yes, in 50 years or maybe 100 years with big data, we will be able to kind of reduce it to the atomic level.
Mhm.
Every single molecular pathway will be completely characterized, and we will be able to perhaps develop treatments based on that. But everything is interconnected, so it's going to be difficult. And I think what kind of differentiate us a little bit as a research perspective is that we are focusing on the bioenergetics. So if you have a car and you ask the question, how can I stop the car from moving? So we would take a look at the gas tank. We would say, oh, maybe if the gas tank is empty, the car will not move. Or if we look at the brakes, if we look at the signaling, okay, if we reduce the signaling to move the car, then the car will not move if we push the brakes. But right now in research, like everything is cancer. So I would say the priorities are not clear. And everybody's trying to chase these trends and trying
Yes.
to, look at everything in the maximum detail possible, which is fine it's producing a lot of data and a lot of information,
Mhm.
but there is only limited time that you can spend on every single thing. So we are looking at cancer focusing purely, I would say, on the bioenergetics, a little bit of biosynthesis too. But so bioenergetics is the ATP. Where is the ATP coming from? And this is, of course, different from the mutations and oncogenes and tumor suppressor genes, locking cancer into a proliferation mode, and then the cell kind of responds to that by reprogramming metabolism. That's kind of traditional perspective. We think that it is the loss of function, or better, insufficiency in oxidative phosphorylation. Maybe we can define these terms a little bit afterwards. So it's the insufficiency or the loss of function in oxidative phosphorylation that leads to an upregulation of substrate level phosphorylation. And this then, of course, it's accompanied by genetic reprogramming as a secondary kind of observation in this atavistic framework where cancer cells revert to this ancient pathway of energy production. And this also accompanied by mitochondrial reactive oxygen species and mutations. So, the question would be, of course, once cancer has developed, we will observe the mutations. So if the mutations are there, what does it matter? What is first, whether it's the chicken or the egg? In the late stage, we will have to deal with both, and that is true. But the approach is trying to target the genes, kind of the genetic product, the gene products. Really, it's we are not fixing really the genes. That gene therapy is not really considered like a very promising avenue in cancer, but we are trying to target the gene products. There is a lot of heterogeneity at when the cancer has already developed, and it's very difficult to target the gene products. So we think that perhaps focusing on the metabolism, there it actually matters whether the mitochondria is functional, how much functional it is, whether it's sufficient or insufficient, how much substrate level phosphorylation, this other pathway of energy production, how much it works. So when we try to target metabolism, we first need to understand, for better or for worse, I would love to only focus on the therapeutic aspects, That's where I think that's where we need to do more work. But at the same time, the understanding of the biology determines how we approach the development of the treatments.
What I think is so interesting, and you're talking about, we've spent the past 100 or so years really looking at this as a genetic disorder, and you are suggesting from your research and other things you've learned that it's a little bit upstream from that, that it's more like the genetics are more of the byproduct of a disorder that's happening at that metabolic mitochondrial level. And so what it sounds like to me is you're also interested, you are recognizing the importance of studying kind of at the bench side, these very singular or even myopic pathways, which are important. We need to have that understanding. But it seems to me, and please correct me if I'm misunderstanding this, you also have a more of a global understanding. Kind of there's like, what does that pathway land within? Like, what is the tumor microenvironment? What is the healthy microenvironment around the tumor, and how all of those interface? And that's where I think you're talking about how do we make this more clinically relevant? Because it's one thing treating it in a petri dish or in a cell line study or in an animal study, but it's different when it actually comes to the patient themselves. One of the things I think is interesting is how would you describe what you just talked about to one of your patients?
Hmm. Well, when I do clinical consultations, I do not shy away from the complexity of the biology. I think it's the language and the metaphors and the examples need to be adjusted for the understanding. But I think it's necessary within the patient autonomy and the scientific literacy. I do think that some basic understanding of the biology is important. Not just mostly also to when you have to have informed consent, you have to understand what exactly are we talking about. So really within like the clinical aspects, I think it's better to focus on not talk about the chondriome and the mitochondrial kind of dysfunction and how the electron transport chain works and all these things, but for the patients to understand more on the side of the clinical kind of the long-term outcomes that come from clinical trials and the different levels of evidence that we have for different therapies, and with the understanding that there are um, certain types of cancer and certain types of diseases where you have to kind of lower your standard for evidence. Of course, it would be ideal if we had phase 3 clinical trials for every single intervention under the sun, but some of these some of these will be quite difficult to do just due to lack of financial incentives to do them. So I think for the patients, it's very important to understand the difference between median overall survival and progression-free survival and the long-term survival. I always try to encourage them to ask as much information for the standard of care that they are being offered as possible, and to understand, they don't have to be statisticians, but they have to understand what these things mean, because sometimes when doing in the standard practice, there's not that much time, and sometimes you hear, oh, we're gonna provide you the maximal benefit, for example, and this means something very different if you have a GBM, if you have a glioblastoma brain cancer of grade 4, the most aggressive, or if you have like a stage 1 prostate cancer or breast cancer. The maximal benefit definition in these two cases is very different. The long-term survival is very different. So I think it's necessary to still go from the biology, because we are fighting two battles, I would say. We have the theory and the therapy, and in the Dr. Seifried lab, we are kind of just in the middle. We are trying to wrestle with both. But I think if we don't understand the biology enough, then we will pursue therapies that have a lack of basis in no biological basis, such as, for example, OXOS inhibitors. I'm sure you've seen the recent clinical trials with this electron transport chain complex 1 inhibitor, has a very long name, IACAS something something, and it led to a lot of toxicity. And I think when they we're testing these things in preclinical systems and in the mice even, sometimes it's easy to forget that there is a human attached to the tumor also. And something that, it would be obvious that if you are going to target oxidative phosphorylation, there will be a lot of toxicity. And even so, even with this kind of very discouraging result in the clinical trial, there's still the idea that there has been a lot of research development kind of investment into this idea of targeting OXPHOS. So there's still a couple of research labs that still continue to pursue this. And even though I think it's not the most sensible thing to pursue, so I think that the biology informs the treatments that you try to develop. And in the mitochondrial metabolic theory, we are looking at the OXPHOS insufficiency and the substrate phosphorylation dependency, and maybe I could define these terms. And this is what for us what drives the malignant transformation.
I love it. And, I think this is a good opportunity because you have talked about, you're alluding to this a lot, but to get really specific and very briefly to define some of these terms that you've been throwing into the conversation here, because it's very, very much to your point, there is a big disagreement on these therapeutic targets and what potential outcomes we can expect. And so when we talk about these targets, I mean, first of all, you talked about this article that just came out this year on this, everything's a target, at complex. And so that's one level that, well, that's never going to get us very far because it's almost too vast, right, of going. And so you talk about some of the targets like oxphos, oxidative phosphorylation. You talk about fatty acid oxidation. You talk about lactate. You talk about amino acid. There's so many of these tools. Can you give maybe a high level overview of what are the biggest targets we're looking at now in the bioenergetics metabolic sphere? First. So let's define that first, and then we'll follow up with what do you personally think might be some therapeutic priorities now and on the horizon.
Yeah, so as you said, within metabolism itself, if you take the perspective that the metabolic reprogramming is a consequence of kind of the gene changes, the gene mutations in oncogenes and tumor suppressor genes, driver mutations, kind of locking the cell into a proliferation mode, and then the cell adapts by upregulating whatever pathways it deems necessary to enable this proliferation. Then there will be a threshold of different things that you can target, which will be different between the normal cells and the tumor cells. And this is true. But you forget, let's say you forgo the fact that you also have the normal cells operating their normal metabolism, and they are sensitive to many of these things, like targeting oxidative phosphorylation, targeting fatty acid oxidation, lactate oxidation, not production, but the oxidation of it, different amino acids. It is possible, and different researchers are pursuing these therapies, and they will provide benefit. But in our mind, this would not be the first step or the first priority. So if I take a step back, maybe I could explain. So we think that each of our cells has a chondriome. So chondriome is the sum of all the mitochondria that we have in any given cell. In our adult tissues, we would have like 100 or 1,000, up to 1,000 of mitochondria.
Are you saying the mitochondrial kingdom is what you're kind of referring to, this like collection, yes, this ecosystem of the mitochondria? Okay, cool.
Yes, I like to think, because sometimes we have in our mind this image of this one giant mitochondria inside the cell, and doing everything. The poor mitochondria is overworked, producing all the energy from the pictures that we have in biology, and that's not the case. So I think it's important, if it's like a functional sum of this 100 or 1,000 different mitochondria, they all have their own mitochondrial DNA. They all have independent function. They travel across the microtubules. They have, it's like a little organism that we have within each of our eukaryotic cells. This is, from the endosymbiotic theory of the mitochondrial origin, like a prokaryotic cell that only could do substrate phosphorylation, engulfed a mitochondria and oxidative alpha proteobacteria. that produces the energy for us right now in the presence of oxygen through oxidative phosphorylation. So if you have this chondrium, this whole kind of functional sum of different mitochondria, they are doing their own thing. Mitophagy, they are being recycled. They are like a dynamic system.
Yeah.
Over time, if these mitochondria suffer chronic damage, and this is not immediate, this is over time, if you destroy all the mitochondria within your cell, the cell usually dies. So it's more a gradual chronic damage. And you can have genetic predispositions for this damage. So, many of these genetic cancer syndromes, such as neurofibromatosis or Li-Fraumeni or Lynch syndrome, these all also, let's say, you have to define the damage, but certainly they lead to substrate level phosphorylation dependency and oxidative insufficiency. And so we would say that the damage or the change in the mitochondria, the metabolic reprogramming driven by the mitochondria, is necessary and sufficient for cancer development and for the sustaining of the cancer phenotype. But the genes, as you know, there is some tumor tissues that have no detectable mutations. So they are there. So they are, in certain circumstances, you could say they are sufficient, but perhaps not necessary, or they are necessary, but perhaps not sufficient, or sometimes they are neither necessary nor sufficient. But the changes in metabolism certainly seem to be present in all cases. If you don't have a cell that is upregulated in substrate level phosphorylation, the common name would be fermentation, but I prefer substrate level phosphorylation because it talks about where the energy is actually produced.
Right.
These two forms of energy determine what the cell will be doing, whether it's oxidizing nutrients or whether it's fermenting them. Now, if we concede that cancer cells have a dependency for substrate level phosphorylation, that is a dependency to upregulate these pathways that produce energy. So that's phosphoglycerol kinase and pyruvate kinase in the glycolysis pathway, and succinyl-CoA ligase in the mitochondria. These are enzymatic reactions that give you ATP, and cancer cells have a dependency to these pathways to obtain their energy, instead of oxidative phosphorylation, which is what you would expect in the normal cell. And now this is where this is kind of our whole thing. That's where we want to target. That's where we want to develop the treatments that will target the substrate level phosphorylation instead of the oxidative phosphorylation. Because yes, there is, and maybe this is something we could touch upon, the counter arguments against the mitochondrial metabolic theory of cancer. And I know this is getting into the weeds and about the biochemistry, but I think it's very important because we never talked about it, and it's kind of difficult because we are trying to convince not only the patients, and we believe the patients will be the driving force to kind of change the system, but at the same time, the clinicians and the researchers, they look at it and they say, well, why are they not talking about the things that are more still in the air? And I think this is something that we have to do. So the counter argument is, of course, that yes, if you have a system where you measure oxygen consumption as a marker of oxidative phosphorylation in cancer cells, you will still be able to detect it. Now, it's very difficult to quantify it and to say exactly how much is contributing to actual ATP production.
Right.
And the efficiency of the system is also very important. So oxidative phosphorylation, as you know, and I'm sure any listener remembering from biology classes, it's a very involved and evolved system. It goes through different steps. So glycolysis gives you a little bit of ATP through substrate level phosphorylation. Then pyruvate enters into the TCA cycle, gives you a little bit of ATP through substrate level phosphorylation. And then this fuels, the reducing equivalents, NADH, FADH, fuel the electron transport chain, the chemiosmotic gradient. And at the end, you get some amount of ATP. You get, the number I learned was 36, no longer. Now it's between 30, 32. And this is because the efficiency can change depending on the system, on the structure of the mitochondria, on whether all the complexes are in their little invaginations in the membrane, on the cardiolipin, on the enzymatic activity of every single enzymes involved in the process of the lipid membrane. All these things determine how much ATP you get. So if we claim that there is a loss of efficiency in the system, of course you still can detect it and you still can measure it, but we think that whatever the number is, it is insufficient to keep the cell alive by oxidative phosphorylation alone. So this is one of the things that this is what I work in the lab. Basically, I have kind of 3 projects. I'm trying to look at try to advance a little bit the clinical side, more of a synthesis of the different ideas and where we could go next from this very, it's not a broad perspective, it's very focused perspective on just the bioenergetics. And then I'm looking at the gravity effect. Well, this is another different thing, but this is
that one will do for another time.
this artifact that you have in vitro systems, that you have how the cell behaves in the presence of glucose, which is very interesting. And then I'm trying to go one by one through the different fuels that either fuel substrate level phosphorylation or they fuel oxidative phosphorylation. And the big question in biology, and many researchers consider this in cancer metabolism, and I've been very happy to see one of the thought leaders in the field also ask the question, isn't it strange that we have been chasing basically a cancer cell that would be able to survive using only oxidative phosphorylation, let's say 90% of its energy, as the normal cells, and we cannot seem to find this cancer cell. And so I'm trying to ask the question and go one by one through all the different nutrients to see if the nutrients that only feed into oxidative phosphorylation can keep the cell either alive, and I think they can, or growing, and here I think they cannot. So if we define cancer as a cell that keeps growing and growing and growing and dividing, then I think the oxphos insufficiency term, where the oxphos is insufficient when you have to upregulate substrate level phosphorylation to keep the system going. And so it's kind of like a circular definition. If the cell is substrate level phosphorylation dependent, that means that oxphos is insufficient. And if oxphos is insufficient, that means that it's either dead or it has to upregulate substrate level phosphorylation. So I'm trying to go one by one through all the different kind of nutrients and metabolites that could give us a clue. And I mean, I'm sure you would think, and of course the logical question is whether this behavior of the cell in cell culture is the same as in the animal. And this is, of course, the answer is yes and no. You often hear, and we hear this as a criticism, that humans are not mice. That is true. Humans are not mice, but both human and mice are eukaryotic mammalian cells, multicellular organisms, and biology is biology. So I think if unless
application across all animals, basically.
You could argue that some species of animals have certain evolutionary adaptations. I don't know. I like the naked mole rat. Maybe you looked exactly. You know about it. It seems to be adapted to hypoxia, and there's something very interesting going on with the Warburg effect. It seems not be able to upregulate the Warburg effect, this organism, because it spends so much time in environments where there's no oxygen that instead of upregulating lactate production, it seems to kind of lower the whole metabolism down. And of course, as you know, they are very resistant to cancer development, although some of them supposedly have been able to develop cancer. So unless we are talking about something very specific, I think most of our eukaryotic cells, our cells in our body, in the minimum requirements for either ATP production or biosynthesis, or the most essential pathways are evolutionarily conserved. It is when you talk about the optimal conditions, most research is developed with, and this is why it's once again, important, the theory and the theoretical background, because when you go back to how the cell culture kind of systems and the cell culture media was developed, Harry Eagle in the 1959, this was the cell culture media was developed trying to maximize growth, because you wanted to have as many cells as possible for
Right,
your
to work on, you have to study on, right?
Exactly. You wanted to have a lot of cells to get a lot of protein and to look at the genes and to try to understand, how all these systems are working in kind of optimal, maximum growth conditions. Now, when you start to strip things away, we skipped all this. Like nobody, well, not nobody, but the purpose of 50 years of research where we kind of ignored metabolism and we focus on the molecular studies, we can assume that this is the baseline, and now everybody will be doing this in the trying to get the cancer cells to grow, instead of trying to focus on what are the minimum requirements for either viability or cell proliferation. So that is kind of, I think a lot of work needs to be done. There has been some work done about it, trying to kind of make a little bit more representative types of media, plasma
Closer to
mass.
what we would expect in the natural form, not in a
Right, in the plasma. The problem is, of course, when you have cells in culture, they are kind of consuming everything that's there, so you have to replenish it, whereas in our blood, it's always flowing. But yeah, I would say in vivo and in vitro, the fundamental constraints are the same.
So a couple recaps, and kind of I would love to clarify, because again, I love this stuff and I could literally eat this for breakfast every day, but I'm imagining there's some listeners here right now going, what the hell is going on? And so let's simplify some things. So first of all, what you're describing is this concept that we're trying to understand the number, the quality, the efficiency of our mitochondria and how it may play into all diseases, not just cancer, but that cancer itself has a unique behavior, different than diabetes, different than cardiovascular disease, Alzheimer's, some of those other conditions that are also metabolically, mitochondrially, disease based, in that there's a shift. You're talking about there is a reprogramming of the resources that those cancer cells take on that's different than, say, the cells in Alzheimer's, cardiovascular, et cetera. And what you're describing here is there becomes a certain dependency that is also not static, that becomes very dynamic, as what I like to describe cancer cells as they're very opportunistic and they are very adaptive. And they will, if you starve one bit of their fuel source, they will find another. These are resilient little buggers and they find their workaround typically. And so you are studying all of those possible workarounds. Is that an oversimplification?
No, it's not. It's true. I've yet to see. So it is true that cancer cells are very adaptable in the sense, and I think there is a danger here to fall to teleology, and I do it all the time in the sense that you have your little cute cells growing in the dish, and they kind of try to scramble different nutrients together as if they had a mind of their own, and they could make decisions. Oh, I don't have enough glucose, I'm going to use glutamine. I think this is not how it is.
Okay.
It's a mechanical response to it's gene determined. It's within the programming of the cell. It tries to respond and adapt to the conditions that it is in. But going through the different nutrients, so there is a limited
being carbohydrates, fats, and proteins, and then a variety of things within each of those categories. Is that what you mean?
correct. But so basically different metabolites that can feed into. All right, so I should give a step back. So there is only two forms. There is only two forms for our eukaryotic cells. We don't have photosynthesis, so we cannot unfortunately generate ATP from the power of the sun. So our cells only can do either substratal phosphorylation and through the mitochondria oxidative phosphorylation. These two pathways can use different metabolites to go through the pathway, but the purpose of you see me falling prey to it. The objective, let's say the end result, not the purpose, the end result of these pathways is the production of ATP. So you can have different
which is the energy currency of the cell.
Correct. So without energy, I think we can agree that there is no life. Nothing can survive indefinitely without energy. You can hang on for a little while, but after a certain period of time, an ATP is very quickly used through the cell. So if you are unable to sustain a continuous production of ATP, of energy, the cell is dead. Okay, so that would be ideal for if we are talking about cancer. Of course, we would try to reduce the amount of ATP, and if the cancer needs more ATP than it's able to generate, the cell is dead, the cancer cell is dead, problem solved. But of course, it's difficult to get there. So you have different metabolites, which come, yes, from carbohydrates, proteins, and fats, the little kind of broken down molecules that feed basically into only these two pathways. Substrate phosphorylation is basically three
one path
points. Sorry?
I was just like reiterating so people understand there's sort of like a fork in the road, and those substrates can go down one particular path or down another.
Yes, the issue is also many of them do both. So that's where the difficulty is. So for example, glucose in normal cells goes through substrate level phosphorylation, and then the majority of it gives you ATP through oxidative phosphorylation. But in cancer, glucose goes mainly through substrate level phosphorylation in the cytoplasm, gives you 2 net ATP, and then it leaves the cell as lactate. And some portion of it also through pyruvate goes into oxidative phosphorylation, but it's a lesser amount. So it's tricky, but some are only for oxidative phosphorylation, such as fatty acids and ketone bodies, for example, and some of the amino acids too. And some are both. For example, glutamine technically goes into substrate level phosphorylation in the mitochondria, in succinyl-CoA ligase, mitochondrial substrate level phosphorylation. And then a portion of it goes also through oxidative phosphorylation, and we think it's less than what you get. So it in the end, it depends on the flux through the pathway. So the more you go through the pathway, and if you have a lot of glucose that goes through glycolysis and a lot of glutamine that goes through glutaminolysis, and it gives you a lot of ATP through the quick way, the substrate level phosphorylation is much quicker, then this is enough to keep the cell alive. And now when you try to force it, so you remove these two fuels, because you can either inhibit them through metabolic inhibitors, or you can remove them, and then you force the cell, okay, now you use only fatty acids, or now you use only ketone bodies, or now you use only certain amino acids. In general, they cannot survive, and certainly they cannot grow. So
yes.
So from this understanding, that's kind of the whole point when you try to develop therapies. We are not saying targeting all the other things doesn't make sense. Eventually, if you prioritize these two pathways first, and you actually make sure you inhibited them correctly, and we would say you have to adapt to the ketone body metabolism. Maybe there we could go into the ketogenic diets and the dietary aspects of it. That is to protect your normal cells. That has not so much to do with the cancer cells. So if you target these two pathways efficiently, so substrate level phosphorylation from glucose glycolysis and glutaminolysis, these are the two main that cancer uses. Then if you see a cancer, surviving cancer cell that is able to oxidize just enough lactate or just enough fatty acids to stay alive without growing, but of course always with the danger that if glucose and glutamine come back into the microenvironment, they will resume proliferation. Then you can figure out, oh, maybe just a touch of an oxphos inhibitor or lactate oxidation inhibitor. That would make sense. But we are now doing the opposite. We are giving you chemotherapy as standard of care, and now we want to give you one extra thing, oh, let's inhibit electron transport chain complex 1, and let's see what happens. And of course, what happens is you get toxicity to the normal cells. So,
so and I think just an important pause moment here of just this, because boy, this goes really fast in these conversations. So I want to bring this back to some clinical relevance for folks. But number one, what you're talking about is how we really could start and simplify things. That would probably be effective, and the literature suggests 70 to 90% of the cases if we start on these two pathways, and then whatever sort of escapes that approach is worth then evaluating, because you can't, no one fits into the one size fits all treatment. And so if we start with kind of the most prioritized, which you just described, of these two possibilities of blocking glucose or blocking glutamine, we will capture the attention of most of those cancer cells, stalling their growth or killing them straight out. And then if there's a workaround, we then go to that as the next priority based on the individual and how they're expressing. So those are really cool. And this is where the studies are. We're getting maybe a little too out there when everyone is trying to target all the pathways all at once. First of all, that's entirely impossible and entirely too toxic and overwhelming for the body.
Yeah, I think they forget there's a patient there too.
Exactly.
You
Exactly.
can target the pathways and it works
Yeah,
great.
you can.
We
You
have
can.
done it, and you can kill cancer cells very easily, but that has been always the kind of the holy
Yeah.
grail
And
of the
sometimes taking the patient along with it, like, great, you don't want to kill the cancer cells and the patient. And this is what's tricky here is like cancer cells are one personality within the body and healthy cells are a different. And so you have to both be supporting the healthy cells while targeting the cancer cells. This is also what a lot of the intellectual bench side forgets, is that there's this other component that needs to be dealt with in real time at the same time, which is why you are so beautiful in this place, because you've got the medical background, the clinical aspect, and the research aspect. And so when you think about today, when you take this information that you're learning in the lab to the bedside, what are for you today maybe your top 3 areas that you're most excited about and most likely would offer to your patients today? And then perhaps talk about very briefly what's on the horizon in these last few minutes together.
Mhm, mhm. Yeah, so I think if we can invest a little bit of the time, maybe the design of the therapies, maybe we could go a little bit into detail, and that would save me also time. So I've limited you probably have much more clinical experience at this point, because I've limited my clinical consultations a little bit, just because I'm so in love and interested in the research that I focus mainly on that. I still do have some time and presence for helping patients, but my focus is I just, out of intellectual curiosity, I'm trying to understand what the optimal kind of therapeutic approach would be, and there is still a little bit of work to do on that front before we would jump to kind of broad statements of every single patient should be treated with this very
Yes.
specific protocol right now. And it's kind of the issue or the danger is that we have a lot of doors open and we are trying to explore them all at the same time, and we get distracted sometimes with kind of projects that are interesting but do not lead to
Right.
helping the patients. And I would say most of the research that is being done at this very moment, unfortunately, it is not done to help the patient. I'm sorry to say that, but
It's true. You're not saying
it's
anything
done to
that's untrue.
it's done to sustain the research itself, and that's fine. I mean, knowledge for knowledge's sake, and I love knowledge, and the more knowledge you have, the more I feel I lack it. But it's
That is
good to have
humbling.
it it's good to have it on PubMed, and I'm sure somebody is working very hard at a very specific transporter that you have in cancer, or on this bacteria, or on this zebrafish, and 50 years from now somebody will Google it, and or whatever the search engine will be at that point, and will say, oh wow, this guy knew 40 years ago that this is the most important thing ever. But for the patient right now, I think we could go through, and this will be in the upcoming publication that I co-authored with Civil, and we have a lot of colleagues from the field on it with us, to try to kind of set up the groundwork for the metabolic treatments from our perspective. And that this is always kind of the case, that our perspective is very focused and it's slightly different from the more conventional perspective, which is fine. I mean, but we only have one life, and we can, I can spend 40, 50 years. Well, hopefully I live a long time, and I can only focus on one thing. So I cannot do everything at the same time. So we would say that the first thing that you would do if you are diagnosed with cancer, you would initiate a ketogenic diet. And when we talk about a ketogenic diet, this has a very specific objective change in the biology. Basically, what you are trying to do is to reduce glycemia and to upregulate ketonemia. And this is like an area under the curve. Over time, you are trying to play your normal cells, which are competing for the resources with the cancer cell, and you are just lowering the substrate availability of the glucose. Especially, you are providing ketone bodies to your normal cells, and you are putting the whole system into like a nutrient scarcity state, like a metabolic pressure on the cancer cells,
Right.
where there is still glucose in the environment, in the
You can't fully
microenvironment.
get rid of it. Right.
Yes, you are not totally getting rid of it. For that, you would need metabolic inhibitors to
Right.
specifically target either glycolysis directly, and 2DG, for example, 2-deoxyglucose is kind of the most famous one, but there is many others. You could also target glutamine with DAWN. Maybe we could also mention it briefly, and or you could do it indirectly with metformin, SGLT2 inhibitors, acarbose, different things that would like just steal the available glucose from the tumor. But you're also lowering mTOR, insulin signaling is very low at its physiological minimum. You're basically putting the whole system into your normal cells are doing good. They have enough for their needs, and you are forcing the cancer cell to, you should be using the oxidative phosphorylation. And that's why it's important that in our mind, OXPHOS is insufficient. It is there and it's being used, but it's insufficient to keep the cell going if you target substrate level phosphorylation. So while you're doing this, that's like the first step. Here, as I mentioned, I would encourage the patients to speak with their oncologist and to get a very clear understanding of what they can expect from the standard of care.
Right.
And this is where I see a lot of failure in our part too, where and I know some patients kind of doesn't want to know, so maybe the family members, but you have to know the long-term outcomes, the long-term 10-year survival of your tumor, so you can make informed decisions whether you should intensify these emerging treatments. They are still evidence-based, but they are still emerging. Or you say, hey, I mean, this is great. I'm just gonna do surgery and one quick round of chemotherapy. Maybe I can do some fasting around it to reduce the toxicity, and the tumor is gone forever. In 95% of the cases, it's not expected to come back. So maybe this is enough. Maybe I don't need to worry about learning what's the chondrioma, what's the substrate level phosphorylation, and all these things. But if I have a GBM and the
Not the
10-year
same story.
survival is 0.8%, then maybe I have to. So after you learn about what's the outcome, and as you know, we focus mainly on GBM, on glioblastoma, which is a model. It's a good model because the metabolism kind of fits very well with these concepts. But at the same time, and I would like to reiterate here that so far from the literature and from my own experiments, we all tumors that we know of, even prostate cancer, okay, so it's going to oxidize some fatty acids. That's great. Is oxidative phosphorylation sufficient to keep it going, and are they dependent on substrate level phosphorylation? And so far, the answer seems to be no in these very specific conditions. So this also mentioned, we always jokingly with Dr. Seifried, we have this prize that we announced of like $100 to $100 to anyone that can find us a cancer cell that would have OXPHOS sufficiency. That is that in the absence of substrate phosphorylation, would be able to live. And I would increase it to, I have the opportunity with you, I would increase it to $1,000. I'll give it from my own money, just to make it more
accepted. No.
just to make it more enticing. People can email me.
I love it.
I'll give them the conditions of the prize. It's very simple. Basically, remove the substrate level phosphorylation metabolites that feed into this. That includes uridine and other things. And if there is a tumor cell that can grow like crazy using only oxidative phosphorylation, I can retire. That's great. I can go back to Spain to write novels, sci-fi novels, and live a happy life. And I don't have to worry about this anymore. So okay, so once you know what type of cancer you are dealing with, then you would bring in the standard of care if it makes sense. In some cases, it makes sense. In other cases, it doesn't make sense. In some cases, there's a little bit of conflict, like radiotherapy for brain cancer, for example. Radiation therapy is great. It kills cancer cells, but at the same time, the brain is just not the
Very good.
anatomical location that you would irradiate because you damage too much the normal cell. So it is a risk-benefit ratio.
Right.
And unfortunately for GBM, it doesn't work anyway. So I would encourage patients to
At least
think
long
about
term. Right,
yeah,
right.
for the long term, exactly. So so it would bring aspects of the standard of care. I think, for example, temozolomide for GBM is great, it's fine. And then you would bring some repurposed drugs, and you know, this probably you have a lot of also like a broad overview of that. And we like mebendazole, for example, but there's many others, you know, doxycycline, the sulfiram, COX-2 inhibitors, different repurposed drugs that are being investigated, and they do not necessarily target metabolism. They can target other pathways as well. Some systemic treatments such as hyperbaric oxygen, IV vitamin C, hypothermia.
Those add extra oxidative stress into that environment. Yep.
Correct. Yes, and they kind of if you are forcing the mitochondria of the cancer cell to oxidize the fuels, and we know it doesn't work as efficiently as it should, and you even provide even more through, you know, hyperbaric oxygen, for example, dichloroacetate, these things, to even force it more to produce reactive oxygen species, you can kill the cancer cells. And then the point would be to bring in the metabolic drugs. And where would you start? So that's the whole point. You would start targeting substratal phosphorylation of glycolysis and substratal phosphorylation of glutaminolysis. And you could do metabolic stratification to try to find out for the specific tumor, you know, in PDX models, in patient-derived xenografts or organoids, you could try to measure as much as you can to, oh, this cell is also very dependent on fatty acid oxidation.
Exactly.
Okay, so we
That's
very
where you
carefully
get a nuanced individual approach to know where some of the workarounds might be.
correct. Although I think if on basic principles, if the targeting of glycolysis and glutaminolysis is sufficient enough, it will stop proliferation. Now, of course, your normal cells also need a little bit of these pathways. It's not like you can just completely remove glucose and glutamine.
We need our healthy cells depend on glutamine. That's the main amino acid they need to survive. And so if you just bring in an across the board glutamine inhibitor, you will take the person out with the tumor. And so you're right. Concepts of press.
Yes and no. So there has been these studies with the pegylated glutaminase that they did together with Dawn. This probably the latest trial with Dawn. So 6-deazo-5 or 6-deazo-5 or 6-deazo-5 oxo-L-noleucine is Dawn. This is this glutamine analog that I'm sure our listeners are familiar with, because we talked about it quite a lot. So that's an inhibitor of glutaminase, but it also inhibits other enzymes for DNA synthesis, purine and pyrimidine synthesis. And it's doing several things, basically inhibiting all the enzymes that are required for glutamine. But when you lower glutamine availability in the plasma acutely to this glutaminase or L-asparaginase, it seems to be fine. The body can tolerate it for the short term, and the same for the Dawn. Dawn administration intermittently,
Exactly.
IV. It's fine. It's more of a chronic. If you instead of pulsing it, if you press it for too long, yes, you will damage the normal cells. And I know this is a point of perhaps not contention, but we talk about Dawn a lot, and I would like to clarify. Dawn is a chemotherapy. It's a research phase chemotherapy. It's a kind of glutamine analog that's in the research kind of field, and we have no vested interest in it. It could be any other glutaminase inhibitor that we could use, and we have no patent on Dawn. It doesn't need to be Dawn. Dawn is just as an example of targeting the pathway. If any other glutamine inhibitor can target the pathway as well, that is fine. I personally would like to try to get a pilot clinical trial with Dawn for GBM specifically. Something very, very small, very easy, easy in quotations. You know, 10 patients, give them a well-measured ketogenic diet where we know in real time, you know, CGM, CKM, we know what the glucose and the ketone bodies are doing actually, and try to measure as much as possible and give them a portion of the standard of care, ideally without radiation therapy, or just delay it a little bit, and then give them Dawn. And I would predict that we would see a very good result. Now, of course, difficulty is Dawn has no patent. So, I mean, we are not against patents. And if changing the molecule and having a patent would entice whatever venture capitalists or investors to help
Invest in
us
the
do the
studies. Right, right.
science, so we could give it to we could apply it to the patients better. I'm open to that. That's just how the game is played. But so these kind of steps will be conceptualized in the paper that should
Yay.
be available quite soon. It's
We're
it's on
excited.
its way, and I think this would be for me the kind of the rational approach to cancer treatment, and it has a lot of flexibility. So that's another thing that it's difficult to wrestle with, is that clinical trials, there's this whole field of personalized medicine in the conventional world, but then you cannot have personalized medicine and then complain that each patient gets something different, and you
Right.
cannot
Right.
translate
You can't have them both. So that's why we have to
yeah,
come up with new models of research and
right.
new translational methods and, different things along those
Yeah,
lines.
I think platform clinical trials would be a way to do it, but they are so expensive. Oh my God.
Exactly.
So there on the clinical trial side, there is a lot of if you, I'm sure
Limitations.
you know about it. If you read about how clinical trials are done, it's all a game. It's all if you have a product and you want to get it to market, and then you get the data later. You
You
first have like
retrofit it to
you know
your desired result that's the nice
exactly
way to put it. And I will say it for you, Tomas, because I know you're in the academic world where you have to probably be careful with what you say, but I'm not. I'm just a savvy consumer and studier of this information. But Tomas, my gosh, there's so much we could keep going on here. But I think big picture is I want folks to be watching for the publication that you and Dr. Seyfried's lab are about ready to publish on this topic. If not, by the time the listeners hear this, that should be out there, which is exciting. I want folks to understand that nothing is set in stone. These are theories. They are not truths, and it is an ongoing process of curiosity and the next iteration, the next step of this. There is also the place of just thinking that there is just one target and one treatment out there. I don't believe that's ever going to be the case, and that we'll continue
we'll
to
have to see.
explore and that we get to continue to learn in this field together. And Dr. Tomas, I feel so grateful of your passion in this because you are helping us iterate to the next level and give us hope on the horizon. So thank you for your gracious time, your brilliant brain, and your beautiful heart.
Thank you so much. And I think we, as we build upon the knowledge that we have, and we as all together as clinician practitioners and researchers in the metabolic treatments field, if we're gonna start agreeing on the most kind of fundamental definitions. From there, hopefully we'll get the incentives to align so that we can actually bring this to the patient. So we need to change a little bit the way we are doing the clinical research. Legitimacy is also very important. So doing the trials in my mind, even though the patients will request these therapies, they will
Before
have to
they're
go
available.
they will have to go through some form of clinical testing, or even just clinical experiences that can be shared as case reports, for example. And I don't think there will be like a one-day revolution where everything will click and suddenly this will be like the new standard of care. This will be more gradual, probably, but hopefully we can get the momentum going, and slowly over time. It angers me a little bit, or it saddens me more than anger, that I'm sure in 50 years time, we will look back, and the people that are kind of making the roadblocks at this moment, from just everybody's trying, and everybody is kind of feeling that we are doing everything we can, but they will say, oh, it was absolutely obvious, this was, it was clear that this was the way, it
Oh
just
yeah.
took some time to get there. So
Yep.
this is kind of recorded, and I want no credit. I will probably contribute like 0.001% to this whole thing, but I think it's dangerous also to feed the ego too much. So I think we should not do that. But just out of curiosity, I really want to try to understand the biology and how this can then be translated into the complexity of the patient, of the human, the whole system, which is where also a lot of the difficulty is. Like you can have very clear mechanisms, and then to bring it into the human is where the rubber hits the road. So
Nice.
this is what we focus on, and sometimes in a like a more combative fashion, I know the new researchers that are coming into the field from the outside. I would like to welcome them to also
Need your
join
help.
us.
Yeah,
Yes, because
yeah.
we are a small lab, David versus Goliath, and it's we cannot do everything. It would be so nice, if I could outsource some of these research questions that I feel are like the most fundamental thing that we could ever ask. And it's only me doing it. It would
I
be
love
great
it.
if there was like 100 other research labs instead of
There's at least
looking at.
one more.
Yes, that is true.
I love it. Yeah,
But instead of spending 20 years looking at this very specific mutation in PhD
right.
kinase or whatever, if we could try to first get a
Yeah.
very good grip on the basic bioenergetics, and then we could move forward with these more kind of specific questions. So that's
Like
kind of
I love it. It's like leaving creating kind of our own chondrium of research would be
yeah,
super cool
that'd be good.
out there too. So let's hold that vision true here. And Doc, thank you so much.
Thank you, Nasha. Thank you so much.